Greetings. I'm happy to welcome you to the 10th Annual Les Turner Symposium on ALS. We have had this scientific symposium for the past 10 years. In the past, Dr. Hande Osdinler has been taking the lead, and she will be speaking today, and we look forward to hearing from her. I'm Robert Kalb. I'm the director of the Les Turner Center at Northwestern University, which is a component of the Les Turner Foundation, which is a large charity that is run out of in Chicago. It's one of the largest independent charitable organization to support the care of ALS patients, the diagnosis and management, the enrollment in clinical trials, and a very strong basic science program that is centered at Northwestern University. The Les Turner Center really works to bring together experts both in clinical medicine, in clinical research, and basic science research. We really feel that this integrated approach is going to be an effective route for bringing new and effective therapies into the treatment of individuals with ALS. So today we will have a morning of basic science talks by leaders throughout the world. And then we will have a plenary presentation by Sabrina Paganini, who will talk about the most recent work on an exciting clinical trial that showed a new drug combination which showed efficacy in patients with ALS. We're excited to hear about that. And then finally, we will have a clinical conversation to answer questions by individuals who are listening to this presentation about some of the more clinical aspects of ALS. Before launching into the research program, I just want to take this opportunity to thank all the people that made this possible, which includes many individuals at the Les Turner Foundation, including Lauren Webb, Jeremy Baldwin, Andrea Pauls-Bachman. In addition, I'd like to thank the Northwestern technical team, Sharon Gladfelter, and also the Meslem Center for Cognitive Neurology and Alzheimer's Disease. And lastly, and truly most importantly, I'd like to thank Telly Moore for all that she has done to make this possible. So now what I'm going to do is give my presentation about what's happening in part of my lab on the basic science end of ALS research. So I'm going to share my screen. So this talk will go for approximately 25 minutes. And the title of my talk is that neurons undergoing pathogenic metabolic reprogramming in models of familial ALS. So the proper function of cells requires that energy production and energy consumption are balanced. There are many energy consuming processes in neurons. There are motor proteins which move cargo around cells. There are enzymatic reactions that use energy, that consume energy. Protein synthesis is an energy consuming process. And perhaps most importantly, in the setting of neurons, is the maintenance of ionic gradients across membranes. So we need to balance energy consuming processes with energy generating processes. And briefly, this includes glycolysis, which generates a substrate, pyruvate, and then acetyl-CoA, which enters into the TCA cycle. The main objective of the TCA cycle is to generate reduced NADH, which enters into the electron transport chain, which generates ATP, which can then be used to feed energy consuming processes. So ATP is really the coin of the realm. It's the energy unit that cells use to power processes. And so the question is, how do we generate ATP, and how do we make sure that the ATP levels that we have in cells is sufficient to drive all the energy consuming processes that neurons require for optimal function. So a very important part of energy production in neurons is the mitochondria. These are, excuse me, these are organelles in cells. You see a picture of one here on an electron microscopy. They're the most efficient ATP generating machines. And as it turns out, there's mitochondrial dysfunction in ALS and in fact, all neurodegenerative diseases. So these are some of the early papers that have, my pointer going here. These are some of the early papers that identified mitochondrial abnormalities in ALS patients, as well as mouse models of ALS that depended on expression of a familial disease that causes, a familial protein which causes ALS mutations in SOD. But as it turns out, it's even wider than that. Abnormalities in mitochondrial function have seen in models that depend on TDP43, another cause of familial ALS, as well as the now famous C9orf72. And perhaps most intriguingly are genes that when mutated will cause ALS, and those genes are specifically mitochondrial proteins. So for instance, this protein CHCHD10, a variety of mutations will cause familial ALS. They'll also, interestingly, cause other neurodegenerative diseases depending on where the mutations are. So CHCHD10 is particularly interesting because we know that it localizes to the mitochondria. It's in the intermitochondrial space. When CHCHD10 is mutated, there's disruption of the function of mitochondria. So this is an intriguing, I'm not going to go into CHCHD10 in great detail, but just to point out that mutations in genes that are primarily functioning in the mitochondria can cause ALS. And so drilling down on what mitochondria do, in particular ATP production, what I'm going to talk about today, is I think going to be common for all forms of ALS, but that remains to be shown. So how can we study mitochondrial dysfunction? So one way is to look at oxygen consumption. So when we isolate mitochondria from the spinal cord of SOD mice, from non-transgenic mice, we can see oxygen consumption as the slope of this line. And if you look at the mitochondria from the mutant SOD mice, the slope of this line is more shallow, indicating that less oxygen is consumed and less ATP is being produced. AMP kinase is the master regulator of energy balance. It controls energy consuming and energy producing processes. And so it has kind of the finger on the pulse of ATP levels in cells. And we have found repeatedly in mouse models, in tissue culture and mouse models of familial ALS, that there is activation of AMP kinase, shown by a few different biochemical measures, and also suppression of the mTOR pathway, which is what one would expect if you wanted to increase ATP production and decrease ATP consuming processes. We also see this in models that involve TDP43. Again, there's an increased amount of phospho-AMP kinase, indicating that AMP kinase is activated in response to a mismatch between ATP production and ATP consumption. So let's just briefly go over the biochemical pathways. Glucose is taken up by neurons. In glycolysis, it is metabolized into pyruvate, which is then under aerobic conditions turned into acetyl-CoA. And the TCA cycle is the main driver for the generation of an electron carrier called NADH, which ultimately generates ATP. So it's these NADHs that are created when the TCA cycle runs, which then get fed into the mitochondrial electron transport chain. These reducing equivalents lead to a proton gradient. The proton gradient then falls through the ATPase to generate ATP. So glycolysis generates acetyl-CoA as well as some ATP. The TCA cycle generates reduced NADH. NADH then feeds electrons into the electron transport chain, which ultimately led to the generation of ATP. So we wanted to study this process in a very simplified model so that we can look to see how substrates are being used over time. And our model involved embryonic rat cortical neurons. There are no glial cells. After 2 weeks in culture, we add viruses that express either wild type or mutant SOD or wild type or mutant TDP43. Again, these are genes which cause familial ALS. We wait 2 days for these genes to be expressed. And then we do something called heavy atom metabolomics. We replace the glucose in the media with a heavy atom glucose, which allows us to see how glucose is being metabolized in cells, where it's actually going. Just to show you that these cultures are really primarily cortical neuron cultures, there are many markers of neurons and very little evidence for the presence of astrocytes. And when we image them, we see lots of evidence for synapses. Okay, so when we do this heavy atom glucose metabolomic investigations of cortical neurons that are expressing familial ALS genes, what do we find? So we are able to track glucose uptake, lactate production. We're able to study the production of the isotope enrichment in components of the TCA cycle. And this is all very, very busy and very complicated. And so I'm going to summarize what it is that we found. So first, we find that there's reduced glucose uptake in mutant protein expressing neurons. Next, we find reduced lactate production in mutant neuron expressing neurons. We also find reduced entry of glucose 6-phosphate into the pentose phosphate pathway, as well as fructose 6-phosphate into the hexosamine biosynthetic pathway. Together, what this means is that there's less glucose entering into glycolysis and less metabolism of glucose through the glycolytic pathway, as well as side pathways. And yet, at the same time, there is maintained or enhanced entry of pyruvate into the TCA cycle in the mutant protein expressing neurons. And finally, there's enhanced production of glutamate, aspartate, and GABA in these mutant protein expressing neurons. So what does this actually mean? What this means is that glucose, which normally enters glycolysis to generate substrates for the TCA cycle, and there are these branch pathways in addition. Under conditions where mutant proteins that cause familial ALS are expressed in neurons, we find a reduction in glucose entry traversing the glycolytic pathways as well as these side pathways. And this is occurring despite the fact that the TCA cycle is working fine. So one question one might ask is, well, if you're getting less substrate through the glycolytic pathway and yet TCA cycle is working normally, what happens to the levels of ATP and what happens to the redox status? So we looked at that very carefully. We looked at AMP levels, reduced and oxidized NADH, reduced and oxidized NADPH, glutathione levels, both the reduced and oxidized levels. And what we find, it's totally normal. So how can we square this? So what we think is happening is that neurons expressing familial ALS causing mutant proteins alter their fuel utilization to maintain ATP levels and redox status. So even though the mutant proteins are likely causing mitochondrial dysfunction, and this leads to an alteration in flux of substrates through glycolysis and side pathways, they are still able to increase NADH levels sufficiently to maintain ATP levels. And at the same time, they maintain redox status. So there's a black box here. It's a little hard to understand how that could exactly work. So to understand that, we wanted to investigate the contribution of individual enzymes in the glycolytic pathway and how they contributed to the remodeling of metabolism. So for this, we took advantage of a yeast growth fitness screen. We took yeast, we engineered them in a way that they inducibly express mutant TDP43. That's indicated by this change in color. And then we just basically say, how fast do you grow? And we find that yeast that have been engineered to express mutant TDP43 have a growth fitness defect. And then we said, well, what happens if we knock out individual genes in the glycolytic pathway? And since there are about 20 glycolytic pathway genes in yeast that are not essential, that is that you can remove them and the yeast will still grow, we could determine whether they have an effect on the growth phenotype, suggesting that they may be important for how yeast are remodeling their metabolism in the setting of mutant TDP43. And in this assay, here are just some examples of what the primary data look like. It's a dilution series. You see less colonies. We did a positive control, which was to look at the loss of DBR1. This was a gene that was found in Aaron Gittler's lab. And we too find that when you delete DBR1, that it suppresses the TDP43 growth fitness phenotype. And then we took this directed screen of glycolytic enzymes. And when you do that, what you find is that several of the genes, when they're deleted, suppress the growth fitness defect incurred by mutant TDP43. And the bars above the line show an improvement in growth. We also found some genes whose deletion made things worse, in particular pyruvate kinase. So this was kind of interesting. And we asked, well, what about other familial ALS or even neurodegenerative disease models in general? So we looked at the effect of our most potent suppressors, YMR, as well as ZWF, which I will tell you about in a moment. We looked at TDP43, DPR toxicity, FUS, TAF, a whole variety of rates. It turns out that these two genes, the loss of these two genes, have a very broad neuroprotective or pro-metabolism effect in models of ALS as well as models of other neurodegenerative diseases. So we were pretty excited about that. So when you look at all of those screens, what you find are 5 genes that come out to be really suppressors of the toxicity and 2 that seem to make it worse. So what are these guys? So again, it's the loss of these 2 genes that is a benefit. And what we find is that this is glucose 6-phosphate. The YMR gene is the mammalian glucose 6-phosphate dehydrogenase gene. And ZWF1 is glutamine fructose 6-phosphate aminotransferase. What are those genes? Well, remember, in glycolysis, we metabolize glucose to generate pyruvate, which can enter the TCA cycle. It turns out that those 2 genes are involved in these branch pathways. And so when you lose these genes, what happens is there is less flux through these side pathways and more substrate going through the glycolysis pathway. And so our interpretation is that anything that improves the flux of glucose through glycolysis is suppressing the metabolic phenotype that we're seeing. So finally, we wanted to undertake some studies in mammalian neurons. So we generated a microRNA to specifically knock down G6PD. When we do that and apply that to our in vitro mammalian neuron toxicity assays, here are spinal cord neurons that are growing on astrocytes. If you express mutant TDP43, we wipe out the neuron population, motor neuron population. But if you knock down G6PD, it turns out that that suppresses the toxicity, and this is shown to be statistically significantly different. So the reason I'm particularly excited about these results is because there are drugs which will target G6PD as well as pyruvate kinase, 2 important genes in the glycolytic pathway. And so I think what we're going to find is that the metabolic dysfunction that appears to be an important contributor to neuron toxicity in familial ALS models, that these are druggable targets. We can actually find small molecules which target them. And I just want to briefly mention some work. These are Agios compounds. This is a company which makes chemicals which bind to and allosterically activate pyruvate kinase. Well, one would anticipate that that would increase the flux through the glycolytic pathway. And in fact, all three of these compounds protect against mutant SOD and mutant TDP43 toxicity. So I believe that metabolic rewiring occurs in neurons that express familial ALS proteins, and that targeting this process can blunt disease phenotypes. And perhaps most importantly, it's druggable. So that's where we're going with this series of observations. I just want to thank all the people that did all this work, Sean Patrick, Yelena, Maria Lim, Jerry Donnell has been really critical in our understanding of what's going on with intermediary metabolism. My longtime collaborators, Itzhak Nissim and Rachel Navy. And this work has been supported by the Public Health Service, the Les Turner Foundation, and the Heather Koster Family Charitable Fund. So thank you for your attention. If there are any questions, now would be a good time to ask them.
Bob, this is Daniela.
Yes.
This was really interesting, and as you know, we've looked at glycolysis as well in our fly models of TDP43 proteinopathy, and although I think our findings are not entirely identical, the focus on glycolysis is really interesting, and we also found that in flies, increasing glycolysis is protective. So I was wondering if I looked for PFK data in your slides, right, the rate limiting enzyme and glycolysis, and it didn't seem to have much of an effect in your case. And I was wondering if you can comment on that. And then second question is whether you tried to supplement pyruvate as an alternative.
So thank you, Daniela. I'm very familiar with your work. I think that one of the things that distinguishes your findings from ours is that we just really have a neuron-only culture system, whereas your work in flies, even with dissected neural cord prep, is probably multiple cell types. And so I think that may underlie some of the differences between what we have seen and you have seen, although in the end, it appears that enhancing glycolytic flux is a good thing. You know, of course, our system is artificial. There aren't the cellular interactions which would normally occur in vivo. We have not specifically looked at pyruvate, except that the compounds from Agios are allosteric pyruvate kinase activators. So because those compounds are known to be safe in human beings and actually are used currently, we are very interested in the possibility that pyruvate kinase may be a reasonable target. You know, as for the three irreversible steps in glycolysis, hexose kinase, phosphofructokinase, and pyruvate kinase, your question is why, what did we see with phosphofructokinase, the rate limiting step? So in the yeast screen, it turns out that there are two yeast pyruvate kinase, phosphofructokinases. And so the fact that we didn't pick phosphofructokinase out in our screen may reflect the fact that there's redundancy among the different isoforms. I really want to try to keep on schedule, and I'm happy to discuss all these issues at another time. But yes, so whether G6PD inhibitors would be effective in other neurodegenerative diseases, the only evidence we have about that was that in our FTD yeast or synuclein yeast knockdown, reducing the abundance of G6PD was a beneficial effect. But again, it's a long way from yeast to nigrostriatal neurons. We shall see. Okay, moving on.